Strategies to mitigate antimicrobial resistance. So the first part of my talk would be on the strategies and the mechanisms of antimicrobial resistance. And then I will focus on what we have been doing regarding antimicrobial resistance and epidemiology of staphylococci in the second part of it, and how we have tried to mitigate the antimicrobial resistance in staphylococci in our lab. So it contains actually totally three parts, I would say. So let me just give you a brief introduction about the antibiotic resistance. So antibiotics have always been considered as one of the most relevant discoveries of the 20th century, but apparently it also corresponded to the rise in the phenomenon of antimicrobial resistance. Which is actually a natural process by bacteria to evolve in such a way to withstand the action of the drugs. So globally, approximately there are 5 million deaths associated with bacterial antimicrobial resistance in 2019. And how do we approach this? I mean, it's not a single approach. We have to have a multifaceted and cohesive approach to tackle this AMR. So any antimicrobial treatments becomes ineffective as infections persist and may spread to others. So there could be various reasons for this, which I will be discussing with you shortly. And this is responsible for millions of deaths worldwide and is considered as major public health concern nowadays. So when we talk about mechanism of antimicrobial resistance, we can say there is a natural and acquired procedure, but mostly it will be acquired through either mutation or gene transfer. And when we talk about gene transfer, vertical or horizontal, but mostly what we see in drug resistance is horizontal gene transfer, wherein conjugation or transformation transduction occurs. But of course, sometimes mutations also gives rise to these kind of antimicrobial resistance. So let us look at the pipeline of antibiotic discovery with the antibiotic resistance. The upper panel of this particular slide is showing the antibiotic introduction or discoveries in various time span. Whereas in the down panel, the lower panel of the diagram shows the resistance to each of these antibiotics in the different ages. So although we consider 1950, the time between 1950 to 1970 as a golden age of antibacterial drugs or antibiotics, but we can actually see the development of antimicrobial resistance. So for example, what I would like to tell you is in the beginning, when the penicillin was discovered in 1943, for about 15 years, there was no antibiotic penicillin resistant strains. Only after 10 years, a decade later, we could see the penicillin resistant microbes. But please, when you look at the scale here, the linezolid in 2000. So this is something, a very important drug. It is a synthetic drug, antibiotic, I would say, but we can see that there is an extensive drug resistance in tuberculosis. So within a span of 1 year, we could also see the linezolid resistant staphylococci. So what I would like to tell you is the rapid development of antimicrobial resistance is giving us a challenge to face the infections in the coming years. So let us just talk a little bit about the antimicrobial drug resistance. Actually, how are they capable to resist this antimicrobial drugs? So there are many ways for the the organisms have evolved to show the drug resistance. So the first example, everyone are aware in this meeting, that enzymatic inactivation. So beta-lactamase is one of the very important enzyme which has to be inactivated if penicillins and cephalosporins have to be active. So what I would say is, now we know that there is a resistance to penicillins and cephalosporins because of the evolving of beta lactamase enzymes. Now it is not one class, but there are several classes of beta lactamase enzyme that has evolved through mutations. And therefore, now this is a challenging aspect for every one of us to look into, especially for the clinical side. So the other mechanism is drug extrusion. So many of these organisms have efflux pumps, which will just pump out all the antibiotics out of the cell. So antibiotic, whatever we are using, has to be in a higher dose so that it has to kill the bacteria. Otherwise, everything, the antibiotic that is actually whatever has gone into the cell will be thrown out of the cell. So one example is with the Pseudomonas aeruginosa and Acinetobacter. So they have developed this kind of a efflux pump resistance. So wherein no antibiotic will stay in the cell, rather it will just come out. Therefore, the bacteria will survive. And also there are the permeability of the membrane is very important. So you can see that if the permeability is not there, the antibiotics cannot get into the cell. Same way, the modification of the drug targets. So if the drug targets are modified due to a point mutation or whatsoever, then again, the bacteria will show antibiotic resistance. So what could be what are the strategies or cutting edge methodologies to manipulate antimicrobial resistance? So this is a very challenging field. So most of the people, students are working on phytochemicals and scientists are working on phytochemicals these days because antibiotics are giving a lot of problem. So either we have to look for a new antibiotic discovery or we have to look for an alternative. But the very promising field is antibiotic adjuvants. And people are looking at intestinal microbiota to enhance the useful microbiota. And apart from this, antivirulence therapy is one strategy what people are looking into because not many people have seen development of resistance here because this is not very crucial for bacterial survival, but rather it changes the state of its living. So quorum sensing inhibitors is again one more aspect what we are also looking into, and many phytochemicals have been uh, adopted for this kind of inhibition. So monoclonal antibodies is of course a very nice therapy. Antimicrobial peptides from the bacteria, other sources, vaccination, phage therapy. Of course, phage therapy has been in the news for a long time, but it has not been translated to the application, and nanoparticles. So with this, there are a number of aspects which we can look into for as a cutting edge methodologies, and I will discuss about a few of them in the next few slides. So as I have explained already, the strategies could be to develop alternative adjuvants to boost the to boost the immune system of the host. Or, there could be two alternative ways here. Or we develop the antibiotics with different scaffold and new mechanism of action. Or we look into the non-essential pathway to minimize the antibiotic resistance. But the very important or the winning strategy would be the combination of drugs, which will actually impact on the synergy. The synergy and the drug combination. So one example, beautiful example, which is there in the literature also is the amoxicillin and clavulanic acid. So this combination of drugs will help the action of the antibiotic in a better way. So you all of you know that amoxicillin is a beta-lactam compound or antibiotic, whereas clavulanic acid, it is from Streptomyces species, and this actually blocks the lactamases, beta lactamases. So when we give it in combination, clavulanic acid will actually block the beta lactamases so that amoxicillin can act on the bacteria. So this is one wonderful strategy which we could be adopting for the future clinical application. So talking about the efflux pumps, there are a number of efflux pumps I would say there are 5 classes, and the 4 classes are common to both gram-positive and gram-negative. So ATP binding cassette, all of you are aware. Then we have SMRs and MFS and the MATE. So efflux pump inhibitors. So the strategy for this is the compounds that we develop should actually interfere with the efflux gene expression so that the efflux pumps are not expressed well, so that whatever compound or whatever molecules we are giving is not extruded from the cell, or adding a functional group to the drugs which can hamper the recognition by the pumps so that they stay within the cell, or interfere with the assembly of channel proteins, or develop small molecules as substrate analogs to block the efflux pump activity. So one example is a reserpin, which inhibit multiple multi-drug transporters like NOR-A. So in the next slide, I have shown you different compounds which inhibit the different transporters. So this is about the efflux pump inhibitors. So you can see on the left side the gram positives and on the right side gram negatives. So here reserpin is one of the very important components for both gram positives and gram negatives. So they try to block the transporter. So once there is a blockage of the transporter, so whatever compound or antibiotics is inside the cell will never come out of the cell, and that will suffice the killing of bacteria. And so that we have a low MIC, so minimal inhibitory concentration of the compounds. Now, coming to beta-lactamase inhibitors, I think most of you are aware of this. So I have already spoke in the first slide. Clavulanic acid with amoxicillin combination. The commercial name is Augmentin. Then avibactam, again, is a combination with clavulanic acid. And this is actually approved for therapy. So, and also relebactam with imipenem. So because clavulanic acid is a secondary metabolite from Streptomyces, it is a beta lactamase inhibitor. So I think one have to keep this in mind, that one compound has to inactivate the enzyme and the drug should act on the bacteria. So antivirulence factors, I was talking about antivirulence factors. So under this, we look into many of the metabolic pathways like sulfur assimilation pathway or quorum sensing pathway. So we all know quorum sensing is a crosstalk between the microbes when they are when they are in the biofilm, and they resist the penetration of antibiotics into them. So by blocking these by blocking the quorum sensing molecules like acyl homoserone lactones or the peptides, we can actually make them sensitize the bacteria to antibiotics. So azithromycin is one macrolide antibiotic which does not have bactericidal activity, but then interfere with the quorum sensing pathway and make them more susceptible for other antibiotics to be acted. So here is one more example of the biosynthetic pathway. So the pathway for the synthesis of cysteine. Usually cysteine is required for many metabolic activities in bacteria, like swarming motility or mycobacterial dormancy. So we want to block the formation of cysteine. So either we can target either of the serine acetyl transferase or acetylserine sulfahydrolase. So now, I have given the example of fluoroalanine derivatives. So this will actually block OASS, and then cysteine is not formed. So if the cysteine is not formed, then the mycobacterium will not go to dormancy, and therefore it is susceptible for antibiotic activity by antibiotics. So here is one particular table which tries to give you the overall view of antibiotic and adjuvants along with their mechanism of action. So just to recap whatever I have said, there are class 1 adjuvants, that is 1A and 1B. One is for active resistance and the other one is for passive resistance. So active resistance, I have already talked about beta-lactamase inhibitors. So suppress antibiotic resistance directly by inactivating the beta-lactamases. And in class 1B, we have efflux pump inhibitors. So all these examples I have already given, carbonyl cyanide, MC207, etc., which inhibit the efflux pump along with a receptorin. And indirectly, they suppress the intrinsic antibiotic resistance. And apart from all this, we also have class 2 that is targeting the host defense mechanism. So if we attach adjuvants to any antibiotics, they can also enhance the immune system by modulating hypoxia-inducible factors. So like vitamin D, that is one example. So we have talked about antimicrobial peptides. So the direct application so far is only the topical administration. The example is gramicidin and polymyxins. And of course, phage therapy is one of the very important aspects because it is highly specific for a specific bacteria, but then it is not translated into positive patient outcomes. So it is trying to give a mixed reactions and mixed outcomes. Therefore, it's not, it has not passed any clinical trials so far. So gene therapy is the advanced therapy with CRISPR-Cas winning the Nobel Prize in 2020 with the editing as a gene editing tool. And this is what we have tried to use in our lab. The story will follow shortly. But before I go to the CRISPR-Cas9, what we have used in our lab, I'll just give you a few aspects of staphylococci because we have adopted this CRISPR-Cas system for our staphylococci to mitigate the antimicrobial resistance in staphylococci. So before concluding this, I would just like to say there is emerging non-antibiotic antimicrobial therapies, which are, as per the World Health Organization reports of 2022, there are 18 new microbial drugs in the pipeline, which includes 46 traditional and 34 non-traditional. Already 34 have are in phase 1 trial, 29 into phase 2 trials, and 14 in phase 3 trial. But already 3 are under regulatory evaluation, and we hope it will come to market at the earliest. And apart from this, monoclonal antibody is proving to be the best aspect for treatment of all these antimicrobial resistant bacteria, especially the escape organisms and Clostridium difficile. So one particular monoclonal antibody is called as tozaxomab, which is actually in the phase 3 trial for Staphylococcus aureus. And if this passes the regulatory evaluation, that would be a boon for many of the patients who are suffering from staphylococcal infections. So with this, I would like to go to the theme of my core subject, that is the epidemiology of staphylococci and why staphylococci is important and what are the different antimicrobial resistance that is present in the staphylococci. So let us just define or try to understand what is epidemiology. So it's actually the process of identifying genetic basis of disease, including variants of the pathogens that influence infection, transmission, and prevention. So Staphylococci is an extremely adaptable and constantly evolving human pathogen. And epidemiological success of Staphylococcus aureus is due to because it's a commensal organism and it has an ability to acquire resistance to newer antibiotics and it has enhanced virulence. So there are both coagulous negative staphylococci and coagulous positive staphylococci. So coagulase negative acts as the reservoir of resistance and fitness genes. That is Staphylococcus epidermidis and hemolyticus. So they are implicated in mild skin to soft tissue infections to highly fatal bacteremia and endocarditis, etc. And also toxic shock syndrome. And there is a repeated vaccine failure for staphylococci because it's changing very often. So here we can see the reemergence of antibiotic resistance in Staphylococcus aureus in the genomic era. And this tries to tell you, the left side tells us the resistance, various resistance that existed and that led to the vancomycin resistance Staphylococcus aureus till 2000 on the left side. But on the right side, it gives the time span of pandemics, hospital-associated MRSA, toxic shock syndrome in US, and the pandemic community-associated methicillin-resistant Staphylococcus aureus. So we can actually understand the timeline of emergence, the timeline of evolution of the antimicrobial resistance in Staphylococcus aureus. Now, drug resistance in staphylococci is mainly acquired by horizontal gene transfer and chromosomal mutations. So I'll be only talking today about the horizontal gene transfer, the events. So methicillin resistance is the most notorious one, and MRSA rate in Indian hospitals is about 41%. And now it has almost increased by another 5%. So treatment options for MRSA is vancomycin, daptomycin, linezolid, although we have seen in some cases linezolid resistance. So molecular basis of methicillin resistance in case of Staphylococcus aureus is SSC-MEC. So this is a cassette, and you can see many of the insertional elements here, IS1272, IS431, etc. So there are many insertional elements. So the combination of CCR gene complex and the MEC gene actually will tell us the different classes of SSC-MEC. So there are about 6 classes of or types of SSC-MEC. Each of them, I mean, we will have to find out what type of SSC-MEC is possessed by the Staphylococcus aureus in order to understand the epidemiology and the spread of the disease. So phenotypic and molecular detection of methicillin resistance is mainly by selecting them on oxacillin and cefoxitin plates, because the MEC-A resistance marks the oxacillin and cefoxitin marks the MEC-A resistance gene. So if MEC-A is present, they are resistant to these two antibiotics. So selection will be made on these plates, and then we look for MEC-A, presence of MEC-A. And once it is confirmed, the second step would be to understand or to know what is the different class of MEC-A, SSC MEC-A, that is present in the staphylococci. So here is an example where we use the multiplex PCR. So the class 1 should give 3 bands with the different kinds of multiplex PCR which have been designed. The primers designed for CCR region as well as the MEC region. And the type 2 should give 5 bands. So here we have you can see the pattern of SSC MEC types in this particular diagram. So I'll just move on to the next one. So this we have done in with our collaboration with our German partner, Wilma Zebua from Würzburg, Germany. So here you can see when we have done the molecular typing with for the MEC cassette, we have found that MEC4, SSC MEC4 types are more prevalent in our Mysore city. Whereas the MRSH, that is the methicillin-resistant Staphylococcus hemolyticus, possessed the class 5 SSC MEC. But class 4 was more predominant, which also suggests that the cassette, the class 4 cassette is a smaller one, and perhaps the transformation or the transduction would be much easier, and therefore the more number of SSC MEC types have been seen in Mysore city, and this we have published in the later stage. So we wanted to do the molecular epidemiology studies, and therefore we did the multi-locus sequence typing for the allelic profile. So why we had to do this multiple locus sequence typing is because we wanted to get the allelic profile and then translate our observation of the SSC MEC, and finally also locate from where these microbes have originated from, or where did it come from, and so on. So epidemiology is basically that we have to locate its original origin. So based on the 7 allele, with the variations of 7 housekeeping genes, we did the MLST profile. So MLST profile, the 7 housekeeping genes are as follows: ARC, ARO, GLP, GMK, PTA, TPI, and YQI. So these are the 7 housekeeping genes which we looked for. So very simple, and we just isolated the genomic DNA and then the PCR for all these genes, and then sequenced it and then sent it to the MLST server, and MLST allocated the different sequence types based on the sequence information. At the same time, it also created the allelic profile and also the geographic distribution of STS. So this is one whole typing method, which will also give us the location geographic distribution of the organisms, or the even when the outbreak occurs. So this is one of the best way to go about it. Now, apart from this, we also did the Staphylococcus protein A typing, that is called as the SPA typing. And Staphylococcus protein A is a 42 kilodalton surface protein. So the amplification was done by PCR because it contains a lot of tandem repeats, and therefore you can see the variable sizes of SPA loci, and this is about approximately 500 base pairs. And these were again sequenced, and then SPA typing was done. So this is the kind of repeats we get, and depending upon that, we get different SPA types like T852, T657, etc. And finally, when we put all the results together, we see here SSC MEC types, the class 4 type, the sequence type, the allelic profile, then the complex, okay, and also the SPA type and number of isolates here. So finally, we could conclude that the different clonal types we have got, and this was generated by eBurst, that is the population for sequence types. And here we can see the STS, that is the founder clones, are shown in blue, and yellow will show the subgroups, and this black color one shows single or double locus variants. So that means from one point, they have emerged or diversified into various lineages of the bacteria. So you can see ST22 somewhere and ST sequence type 8 somewhere else. So that means there is a lot of divergence of these organisms with the time. So evolution has occurred, and finally they have emerged into various kinds of lineages. So finally, we try to publish this data in Infection Genetics Evolution. And my student, Dr. Vineet, he did a very good job, and we were able to publish this in a good impact factor journal. So this is how the allelic profile was generated. And we could also see where our clones were, which were circulating also in Mysore. So we were quite amused to see this ST8, which is a US type 300. And we could also see that it was located in USA and also many of the other regions in the all over the globe. So this tries to tell us how the bacteria can be transported through either through the people when we are when we are traveling, or through travelers, or through the export of any of the materials. So now coming to, so when there is such a widespread of bacterial infections or viral infections or whatsoever, so this study of epidemiology will help us, multilocus typing or whatever, will help us to understand not only the antibiotic resistance, at the same time, it will also give us an overall picture of the distribution of these lineages all over the world. So with this, I would like to say that when we have seen so much of antibiotic resistance, we wanted to see how should we really look into or combat this particular aspect. So with that, we tried to look into this CRISPR-Cas system, because one of my students was very much interested into this, because this is a hot topic. This was a hot topic at that time. So we all know it's a genome editing tool for characterizing the genes that is responsible for virulence and also drug resistance. So what is CRISPR? It's a cluster regularly interspersed short palindromic repeat with CRISPR-associated protein, that is the Cas protein. And this is actually a diverse prokaryotic immune system. That means this is present in prokaryotic system as to mark the immune response. So it gives immunity to the bacteria in order to defend against any invading DNA or genetic element. So CRISPR-Cas systems are of two broad classes, and there are 6 subtypes, but I will not go to the subtypes, but I'll just talk about the 2 broad classes. So please look at the upper panel here. The class 1 has a number of effector proteins. You can see Cas5, Cas6, Cas1, Cas7, etc., Cas8 and Cas3. But whereas class 2 has only single effector protein, either Cas9 or Cas12 or Cas13. So for our experiments, we have used Cas9 as single effector protein in the coming experiments, in the following experiments. So I'll just explain about how CRISPR-Cas acts. So genome editing using CRISPR-Cas system. So CRISPR loci, it is a small, short, repetitive invader-derived sequence called as spaces. They are integrated between the similarly sized DNA repeats, and these spaces are transcribed to form the CRISPR RNA. And protospacers, they contain PAM, so it is different from the spacer. So PAM sequences are about 2 to 6 base pairs, which are just beside the protospacer. And tracer RNA is a trans-acting CRISPR RNA, which is a non-coding RNA, which form a complex with the spacer which transcribe the cRNA to guide the Cas protein to cleave the DNA. So basically what happens is the repeat spacers. So this repeat spacers produces the cRNA, which will specify a single target for destruction, while the tracer, that is the trans RNA, cRNA, it's a short RNA. It forms a complex with the single guide RNA and single strand guided RNA and recruits CAS protein, that is the Cas-associated protein for editing. And the Cas9 protein has an RNA guided endonuclease activity, and therefore it cleaves the target DNA. So this is how it looks. So this is the spacer. So spacer makes the cRNA, and then this will be associated with the trans RNA, and this is the protospacer and the PAM. So it has to be recognized, and the sgRNA and the cRNA will combine to recruit the Cas9 here, and endonuclease activity will occur at the protospacer. Now, the strategy what we have used. So please give a minute of thought here. So we have used 2 types, that is Cas9 and dCas9. So what is Cas9? So Cas9 protein has an endonuclease activity. Wild type Cas9 has an endonuclease activity, and dCas9 is catalytically inactive protein. That is, it does not have any endonuclease activity. So we have tried to use both the strategies here. So the wild type Cas9 endonuclease activity, the property is such that it will bind along with the CRISPR RNA at the PAM region with the protospacers here and cleaves the target. So when there is a cleavage, there is no transcription and no translation. Therefore, that protein is not expressed and okay, so that is the mechanism. Therefore, the bacteria gets killed. I will talk about how bacteria is killed with the targeted cleavage is in the next slide. So now talking about the dCas9, which is catalytically inactive, it just recognizes the site which is just next to the PAM, but then it cannot produce endonuclease, therefore no cleavage, but rather stalls the RNA polymerase. So therefore, no transcription. So the transcription is hampered. Therefore, no protein is produced. Rather, the gene is silenced here. So gene silencing occurs with the dCas9 protein, and no transcription at all with the Cas9. So these are the two different plasmids which we have used in order to understand the mechanism, their mechanism in Staphylococci species, which are methicillin resistance. So PCAS-A will have the endonuclease activity and PSD1 does not have endonuclease activity. So we evaluated, we call these plasmids as antibacterial drone. So antibacterial drone CRISPR-Cas9 for bactericidal efficiency on Staphylococcus aureus. So this antibacterial drone, which we constructed with the plasmid Cas-A. So we just put the empty plasmid P-Cas-SA to the Staphylococcus Newman strain, and we could see that there was no activity because there is no target in our plasmid. Whereas in case of the second picture here, we put the target gene, that is the MECA spacer. Therefore, the CRISPR-Cas9 did the job of endonuclease. So it cut wherever the MECA gene was there, and therefore no bacteria survived here. Whereas here, in case of PKSA AGRA spacer, because we also targeted one more gene, AGRA, which also gives resistance, so the bacteria was completely killed. So there was no trace of any bacteria. So efficient killing was done by ABD, that is the antibacterial drone CRISPR-Cas9. So we could not recover any bacteria from this. Therefore, we tried with the dCas9. So when we have introduced dCas9 to MECA clones, so we can see here, there is a silencing for a longer time, but later, after 6 hours, there is a small growth, but definitely it will not reach the maximum growth. So here, because there is a silencing for some time, and with the methicillin pressure, they finally try to, the silence gene will start reverting back to the normal phase of growth. So it's not it is like bacteriostatic rather than bactericidal. But on a normal media with the PSD1, the MECA will grow, but here there is a selection pressure of methicillin. Therefore, it cannot grow because the transcription does not take place of MECA genes, and therefore they are kind of resting here for about 6 hours, then slowly grows, but still it will not reach the required growth rate to cause the disease. So selective media is MRSA, and extended lag phase and significantly reduced OD indicate effectiveness of MECA dCas9 module in inhibiting the growth in the presence of methicillin. So this is the take-home message. So we can either use dCas9 or Cas9 to control the bacterial antimicrobial resistance in case of Staphylococcus. This is just an observation, but a long way to go in this aspect. So that's why we also looked into the transcriptional, other gene expressions in different clones like AGR clones with dCas, MECA clones with dCas, and SPA clones with dCas. So when we have looked into this, this is very interesting aspect here. With AGR, whenever we have a spacer, AGR spacers, so there was many genes were downregulated. MECA was downregulated, HLA was downregulated, and SPA was downregulated in AGR. But in case of MECA and SPA clones, only the SPA gene was downregulated, only the MECA was downregulated. So what we thought was the AGR spacer would be an optimal spacer to use in order to control the antimicrobial resistance. So because of the previous result here, hemolysin completely downregulated. So we try to see the hemolytic activity. So we try to look at the hemolytic activity, that is because the physical activity. So now when we did the alpha hemolytic assay, In case of PSD1 DCAS9 mediated disruption of AGRA gene, we see in wild type there is always hemolysis. Where there is only the plasmid, there is hemolysis. But when we have put the AGRA, hemolytic activity is completely controlled. It is completely gone. So this actually fortifies our observation of different gene expressions in DCAS9 with AGR spacer. So to recap, the molecular epidemiology of MRSA, the global spread of MRSA includes very distinct transmission dynamics and risk factors. So to understand the genetic basis of resistance, especially the MEK-A gene and various SSC-MEK, it's very crucial for tracking and managing MRSA. And CRISPR-Cas and DCAS treatment can effectively target antibiotic resistance genes, offering a novel approach. But DCAS, as we have seen, DCAS can bring about the silencing of gene, providing a tool for controlling bacterial virulence factors without killing the bacteria directly. But future research and clinical applications are that we have to focus on to improve the delivery mechanisms of CRISPR components, enhancing the specificity and minimizing the off-target effects. And of course, future clinical trials is essential to validate the safety and efficacy of CRISPR-based treatments for MRSA diseases in humans. And definitely, we have to also look into the animal model to look into the CRISPR-Cas aspect. So these are my contributors. Mr. Vineet has worked mainly on molecular typing and epidemiological studies of the staphylococci, and Preeti has worked on the CRISPR-Cas system, and she's going to shortly submit. And these are my two collaborators from University of Würzburg, Germany, with whom long-standing association of Vineet, myself, and my former student have been together. And I thank all of them, and I thank you for your